So while Aki's setting up, I'm gonna already can give the short introduction. Aki is currently a research scientist at the Child Mind Institute in Midtown Manhattan, and he's interested in brain development, skill learning, reproducible and open neuroimaging methods. And he will give us a nice presentation about bagging improves the reproducibility of functional parcellation of the human brain. And with that, I would say, Aki, you go.
Thank you. Thank you. This is a really, really amazing thing that is happening right now. I'm very excited to be a part of it. I have been introduced to the open science community kind of through Nilearn. And so this was a really, really just, this is a really exciting thing for me to get to participate in. Today I'm going to talk mostly about this some recent work that I have been doing for the past couple of years, and then at the end I'm going to sort of talk about a little bit about more of my some of the work I do with Nilearn and teaching Nilearn to people and things like that. So here's the outline for what I'm going to talk about today. So I'm going to sort of briefly discuss functional parcellation and why reproducibility is such an important concept when we think about functional parcellation. I'm going to discuss this thing called bagging, Bootstrap aggregation, and then get into just very briefly mention this Python package that I developed called PyBask, which is an application of bagging for clustering. I'm going to tell you about how in this study, the bulk of the talk today, how we looked into how you could use bagging to improve the reproducibility of parcellations. And then I'm going to finally wrap up just with a little discussion about my work teaching Python and Nilearn to the public and community members, non-scientists, and some scientists in a public forum called GenSpace, which is like a community biohacker space in Brooklyn, New York. So just to begin, I just want to start with some acknowledgements. Probably first and foremost, the open science community, because I wouldn't have been interested in doing any kind of work like this were it not for my introduction through the NeuroHack Academy and the OHBM Brain Hack and all the work that the Open Science Special Interest Group for OHBM does. I think I coded in MATLAB throughout grad school, and when I switched over to my postdoc at the Child Mind Institute, I got very heavily involved in Python, and I made my first pull request, I think, on some documentation, Nilearn documentation in 2016, and it's been it's been a nice ride since then in this Python and open science community. I want to thank my co-authors, Ting Zhu, Annabelle Sloan Heinsfeld, Pierre Bellick, Joshua Vogelstein, and Mike Millen, and my funding sources, the National Institute of Mental Health and the Brain and Behavior Research Foundation. So, functional parcellation. It's a very important topic, obviously, in functional neuroimaging. It's one of the sort of key linchpin frameworks which we think about brain organization, and it can be conducted with a variety of different approaches. So we have things like global similarity-based clustering. That would be sort of your more typical sort of thought, what you might think about in terms of clustering based on the entire connectome or spatial similarity of connections or the time series of all voxels in the brain. Or it might be something like a multivariate decomposition into a suite of different sort of components. Or it could be an approach that uses some kind of local gradient transition, where it's looking for local changes in the connectivity of a given vertex compared to other regions, and sort of making boundaries that way. So there's a lot of different ways to do it. It is probably one of the most like fervently studied areas right now of how to understand the connectome. But there is a really important sort of need to understand reproducibility within the context of functional parcellation, and perhaps more broadly in the context of clustering as well. So, you know, functional parcels are sort of the basis of any further graph theory or network analysis of the brain. So if you just think about all the studies that are conducting sort of modularity-based analysis or hub-based analyses or anything like that, those analyses of the functional connectome are all contingent on the definition of the atlas. So you know, graphs and graph theory, there's only really two components, right? There's nodes and there's edges. And everyone, you know, we spend a lot of time sort of mapping out how these edges vary with respect to, say, age or diagnosis or something else. But the node definition is sort of like the first definition that happens. So depending on how you define your nodes, your edges may be actually mapped quite differently. And we see, for example, that, like, you know, generally speaking, across a lot of different types of atlases, we don't see actually very high spatial consistency between them. Because a lot of this has to do with how the atlases are built. Some atlases are built, like I said, with multivariate decomposition. Some are built with local gradient-based methods. Some have very strong spatial constraints. Some have less spatial constraints. All of those things are going to differentiate how the atlas ends up looking in the end, right? How that atlas represents the functional connectome. Not that, but so all these atlas are generally using different data sets. And there are going to be scanner effects for each different data set. There are going to be important considerations to think about with regards to which population was used to make one atlas versus another. And another aspect is that if the functional connectome itself is not really reliable, if the edges of the connectome itself are not really reliable until, you know, maybe you exceed 30 minutes of fMRI data, that could also indicate that our application of parcellations to 5-minute or 10-minute data might not be very robust or reliable either. And the final point is just this was nicely demonstrated by Janine Bister-Bosch a couple of years ago, showing that most of the variance in the edges of a connectome can be explained based on the alignment of that atlas to that individual's fMRI data, right? So the implications of that are that if we are applying different atlases to an individual person, we may be conflating differences between individuals based on their, say, age or diagnosis or other sort of cognitive or other status with the degree to which that connector or that atlas is fitting that person, right? So that atlas may not actually be an accurate representation of that of the sort of functionally homogeneous regions of an individual's brain. So I just want to talk a little bit about reproducibility because sort of it's discussed in a lot of different contexts, right? I think probably most commonly we discuss something called computational reproducibility, which is something that, you know, I can run something and you can run something. Like if I have a sort of set of analyses, we both can run that set of analysis. I'm going to discuss reproducibility from a different perspective here, and I'm using this definition from the Joint Guide for Conditions of Measurement. If you are really interested in reproducibility, terminology, all that stuff, you know, this is like coming from the group of people who sort of define what the official weight of a kilogram is and that kind of thing. So reproducibility is defined as the similarity of results of the same or similar measurements. A measurand is like the thing being measured, carried out under changed conditions of measurement. So the thing about reproducibility is that you have to specify what are the changed conditions of measurement, and that is the reproducibility that you're seeing. So for example, if we have change conditions of measurement across different scans, then that's between scan reproducibility. If we have different sessions, so take person out of the scanner, right, and then they come back later, maybe another day, then that's between session reproducibility. If we have between sample, then that might be two groups of people, and we look at what the parcellations look like between those two groups of people if we were to conduct a functional parcellation on both groups and then assess the similarity of those functional parcellations. And then we have reproducibility across studies. So if we have one group of one study over here with a set of functional data and another study over here with a different set of functional data, how similar are those two studies in terms of their functional parcellation? And then finally, populations being like, are the underlying sort of assumptions about the type of people who are being scanned different in some important way? So there are a lot of different applications of Bootstrap aggregation in the literature, most of which focus on methods that can assist in prediction. So you might be most familiar with Bootstrap aggregation in the context of random forest or something like that, right? It's a set of ensemble methods. And there's an element that we think would be applicable to functional parcellation and bagging, in that there's an important element that the sample that you ended up getting, right? Not only the sample in terms of the subjects, but also the sample in terms of the time series that you ended up sampling are random. So the people who ended your study and the people who ended up or the hour of the day or what they were thinking about at that exact moment are somehow random, right? And so we don't want to, when we're conducting a parcellation, to be actually saying that this clustering is we want this clustering to accurately represent exactly what was happening in these people's heads at that exact moment in time. We want to somehow get an assessment of like the variability in both the sample in terms of the individual and in terms of the time series. And so this bagging approach, and I'm going to show it in a more concrete example in terms of neuroimaging, but generally works like this. So we have a data set and we create some kind of resampling step that enables us to create multiple data sets from that original data set, and we perform a clustering on each of those data sets. And then there's some kind of consensus aggregation across all of those different clusterings, which then gives us our final clustering in the end. So the first half on the left-hand side is our, I'm showing with my mouse, is the first half on the left-hand side is our bootstrapping, and the right-hand side is our aggregation, and that's where we get this bootstrap aggregation. And so PyBask is a Python-based implementation of an extension of this bootstrap analysis of stable clusters method that was developed by Pierre Belek and shown in 2010. And I'm going to briefly describe how this kind of approach works. But in order to do that, I'm going to first describe a sort of a more standard approach at how we might look at clustering. So, for example, we have in the top here, the standard approach, we have different subjects, subject 1, 2 to subject n. And we take their pre-processed fMRI data for the whole brain, and then we represent it in some kind of super voxel resolution, right? So the brain has tens of thousands of voxels, and it's computationally very demanding to be able to actually perform clustering on such a large matrix. So we downsample it to something more like in the range of thousands instead of tens of thousands. And then from each of those super voxels, we might extract, we would extract a time series. That's the average of all the voxels in that super voxel. And the super voxel can be created with a variety of different methods. People can use some kind of hierarchical feature agglomeration. That's what we do here in with using Nilearn. And then you know, a time series is extracted, and for every supervoxel, and a clustering is applied, and then that clustering is represented here in this thing called the adjacency matrix. So this adjacency matrix is a supervoxel by supervoxel sized matrix, which has a 1 if 2 voxels are in the same cluster, or a 0, or sorry, if 2 supervoxels are in the same cluster, or a 0 if they are in a different cluster. And then those individual adjacency matrices all get aggregated together to create a final group adjacency matrix, which is then itself submitted to a final clustering to get sort of what the final parcellation might look like. In the case of bagging, there are two different steps that bagging is happening now. So if it was just more sort of standard bagging procedure, we would just be bagging across individuals. But since we have this, we have time series, and we think that we could probably show benefit from bagging the time series as well, we do it at this multi-level bagging approach. So the first step is, in the same regard, you take the individual's data, you create this supervoxel resolution, and then you use Bootstrap aggregation, or bootstrapping rather, first to resample that subject's 4D time series into a set of, say, 100 or 200, or even as little as 5 or 10 other time series. So we take from one 4D matrix now to like X 4D matrices, where X is the number of bagging bootstraps that we're doing. And then each of those resampled 4D matrices are then, and I should just mention that the type of bootstrapping here that is done is called a circular block bootstrap. So it's taking pieces, contiguous pieces of the time series, and concatenating them together. And the pieces that are taken are themselves chosen at random. And so we have these multiple time series, and then each of these 4D images gets clustered, and we get an adjacency matrix for each bootstrap. Okay, and then those adjacency matrices, for this still now only at the individual level, those adjacency matrices for the individual subject then all get aggregated together into this mean individual adjacency matrix, right? So whereas the adjacency matrices themselves, they are binary. They're ones and zeros. Ones where when 2 voxels got clustered together, zeros when they weren't clustered together. Now they get aggregated into this with this mean adjacency matrix. So now what we're seeing is the proportion. So each cell now represents the proportion of times across bootstraps that that 2 voxels were clustered together, right? So if we have 100, bootstraps, then we would have some value between like 0 and 100 that described how many times the data were clustered together. And then at this second stage, so I'll just point out that the red arrows all here show the bootstrapping part, and then the blue arrows show the aggregation part. So this next part here is all these red arrows are showing the bootstrapping part. So now, but the bootstrapping part now at the group level. So now we're aggregating not, now we're bootstrapping not the time series, but now we're bootstrapping which of these mean adjacency matrices get included into a group, a bagged group adjacency matrix, right? So now we have different groups of individuals. So this one has some proportion or mixture of the individuals in our sample. This one has another proportion of individuals in our sample, et cetera. They all get aggregated together into this mean bagged group clustering. And then then that final clustering is then, or that final adjacency matrix or stability matrix is then clustered to produce the final parcellation. All of this in PyBask is done with the help of Nilearn and scikit-learn and NiPipe, which are all really wonderful tools. I'm very happy to have gotten to use them. So a little bit about the samples that we use in this study to sort of assess whether we can really improve the reproducibility of functional parcellations using bagging. We used two data sets. One, the Hangzhou Normal University data set, which is a fantastic stellar data set if you are interested in reproducibility. It's 30 subjects. They are each scanned 10 times for 10 minutes per scan. They are all in their mid-20s and 50% male, 50% female. And we have the genome superstruct project. The HNU data set was collected in China, in China, and the GSP data set was collected at Harvard by Randy Buckner's group. And Avron Holmes is the first author on that. So, and Xingyan Zuo is the first author on the HNU core initiative paper. This GSP has 300 subjects. Each of them are scanned for 2 6-minute scans. So this is within session. So they have two scans in the same scanner session. Whereas in the HNU, it's 1 scan across 10 sessions. So this enables us to look at both between session effects and between scan effects. And the large sample of the GSP data set allows us to look at within population, within study, between sample effects, and compare actually these two. So I'm going to go over a little bit of different types of generalization that you can think about when you're when you're talking about reproducibility in this context, and that we studied specifically in this study. So first, and I put these little blue circles and numbers here so that you can kind of keep track as we go through the figures. You can kind of keep track of which level of reproducibility we're looking for. So generally, this 1 is the closest reproducibility, and then this 5 is the furthest away, right? So between scan, they're the same population, same sample, same scanner, but the data is different because it was just acquired, several minutes apart. So we have this GSP 10 group test retest, so the 2 scans. Then we have, for 2, we have this between session, which is same population, same scanner, same data, again, but now we're looking at the HNU because now we have 2 sessions, or at least 2 sessions per person. So we have 10 per person actually here. And we have reliability, where we are actually looking at the relationship between the variance within an individual across sessions and scans, and to the variation between individuals. And then down here at 4, we have within study between samples. So this one's kind of a mouthful. So within study means that we're looking at the same study. So we're still looking at the GSP, but we have between samples. So we have 300 subjects. So now we've split those 300 subjects into 10 groups of 30, right? To keep them consistent with the size of the HNU data set. And so now we have the same population, but now different sample, different scanners, and different data that are acquired because the GSP was acquired in multiple scanners. And then finally, we have between study, between sample. And this is sort of the furthest level of reproducibility that you can imagine. And ideally, the one that you want to, that one that we are hoping for the most, right? Because if we can't get, if we have between study and between sample reproducibility, then we feel pretty confident that we can sort of say that this method would help harmonize results across different data sets, not just different data sets that are acquired within a single study. So the approach that we took here was we looked at both the question of cortical parcellation and subcortical parcellation, both in parallel, because while cortical parcellation sort of gets the sort of majority of attention and effort, the subcortical parcellation is also really important, and is actually a particular interest of mine. Subcortical areas and cortical striatal interactions are a particular area I'm interested in, and they also are an area of the brain that have typically less, well, people think at least have less signal to noise compared to cortex. That's mostly because of head coils, but there also may be endogenous elements of how subcortical areas are connected to the rest of the connectome that drive variance in their connectivity and time series that make them look less reliable. So we wanted to sort of demonstrate that this parcellation reproducibility element would be consistent regardless of which area you were looking at. And then the sort of overall picture here is to look at the impact of bagging on parcellation reproducibility, and how that effective bagging interacts with the length of the scan time. So how much scan time do you have in your scan? What parcellation target, where are you parcellating, and which number of how many bootstrap aggregations are you actually using? Because as you use more and more bootstrap aggregations, it becomes more computationally demanding. So the first result, and I guess I should maybe pause for just in case there are questions. I'm not seeing any. Are there questions? I see. Gael, should we believe that there are indeed functionally homogeneous regions of an individual brain given the complexity of the brain? No, I don't really think that there are, but that's not going to stop people from trying to analyze the brain. I think there are, we can say something like this. There may, given that a lot of people, and I'm going to talk about this sort of at the end as well, but given that a lot of people are focused on discussing the sort of variability in the time series and variability of connectivity, we shouldn't necessarily believe that there's anything like a strict atlas in terms of what a region of the brain should really be. But there may be some kind of central tendencies that may exist. And personally, I tend to believe that perhaps rather than maybe looking at from this perspective of the entire brain, maybe breaking it up into sort of smaller interregional parcellations or between network parcellations might be more realistic. For example, we might not be able to understand what would be the best approach to parcellate the, say, the paracingulate gyrus or the precuneus, but maybe with respect to how the default mode and the frontoparietal network are related, or the singular-percular network are related, there may be some kind of more dominant consistencies there. Okay, going back to this. So, okay, so this is our sort of first stab approach. So we're looking now at between scan reproducibility. And so on the left-hand side, we're going to see a lot of these four panel figures. So I'm just going to use this one to sort of introduce how they're shaped. So on the top here, what we're looking at is the spatial correlation of those adjacency matrices. So how similar is the voxel by voxel relationship in terms of the parcellation. And on the left-hand side, we're always going to have the non-bagged condition. And then after the dotted blue line, we're going to see what it looks like with 10 bootstraps, 100, 200, and 400. So, and then generally we'll have for these, this one is subcortical data, and this is looking just at 6 minutes. So 6 minutes is like on the lower end of the amount of time that we ended up investigating. We go up to 50 minutes at the upper end. And then we have two different parcellation resolutions. We have K10, so 10 subcortical clusters, and then K18 is 18 subcortical clusters. And then this second metric down here is called the adjusted RAND index. And so this is a sort of a permutation free method of measuring how similar the voxel cluster assignments are. So for example, if I were to measure two parcellations, one with a cluster label of 1, 2, 1, and the other with a cluster label of 2, 1, 2, the adjusted rand index, since it doesn't care about these permutations, it just matters about the overall pattern, it would see those as identical. That would get an adjusted rand index of 1. So we see generally that bagging improves the between scan reproducibility of our parcellations. And then here we're taking a look at cortical parcellation. So this is 2. Now we're looking at between session reproducibility, and we're looking at the reproducibility from 7, 20, 197, and 444 clusters. I think one thing that we should note specifically is that generally we see a decrease in the consistency of our parcellations as we go into higher orders of regions. This is only based on 10 minutes of data. Again, no atlas that really people are using was created on 10 minutes of data, but this was sort of like a proof of concept, more or less, to demonstrate that even with 10 minutes of data, the Bootstrap aggregation would improve. So we see generally that we are improving the reproducibility, and I think one notable thing to look at is, so here the red ones are the non-bootstrap condition, and the teal are the bootstrap condition. One thing worth noting is that the reproducibility at the level of sort of what we would typically call like a network, which is like a 7 parcel resolution, is like what we see as the highest, and we can and since as we go down into finer resolutions, the reproducibility also drops. It's a nice thing to see that the bagging at 197 clusters can give us something that can be about on par with what we would get with a 7 cluster resolution, suggesting that bagging can be one ingredient that can help us investigate more finer resolution organization of the brain. So this is now looking at, so that the previous slide I showed you was with 10 minutes of data, and now we're looking at a range of different amounts of data. So from 3 minutes, 5 minutes, 10 minutes, 20 minutes, 20. And we're showing that same pattern here. So now this is looking at the subcortical parcellation. In particular, another reason that we did this cortical and subcortical parcellation was that that was pretty nice, was that since the subcortical parcellation was so much smaller, we could run many more conditions with the same amount of cost on Amazon. So it enabled us to like explore the search space of the effect of bagging more broadly. So we show for spatial correlation, we show that if you look at that adjacency matrix, even 3 minutes of data shows very dramatic improvement in terms of reproducibility, even with only 10 bootstraps, right? So as we go into higher and higher bootstraps, we see, and we get up to 50 minutes of data, we almost see a perfect correlation between the two, between multiple sessions. In the case of, there are 10 scans here that are spaced apart on an average of 3 days. To create the 50-minute scan, what we did is we aggregated all of the odd sessions, and then to create the reference, we aggregated all of the even sessions. And all of these 3-minute, 5-minute, 10-minute, et cetera, they're all compared against that same 50-minute reference that we're calling our gold standard, right? So as you see, as you get into higher amounts of data, you get almost a perfect alignment. And we see that this pattern is the case for the spatial correlation for both K10 and K18. And the adjusted rand index shows a similar story, except that it doesn't actually seem to improve the parcellation at the very lowest amounts of data. So for 3 and 5 minutes, we don't see as much improvement as we do for, say, 10, 15, 20, et cetera. One thing that's really worth noting here also is that the bagging is improving the reproducibility of the parcellation more in some cases than doubling the scan time, right? So we can see, for example, going from 15 minutes to 30 minutes in the non-bagged condition leads to a smaller improvement than going from 15 minutes, going from 0 bootstraps to 400 or 100 bootstraps, for example, in 20 minutes. So if you look here, for example, you'll see that from 20 minutes to 50 minutes, we see this increase, but from this 20 minutes to this 20 minutes, we see a greater increase with bagging, right? So this is suggesting that bagging could be a way for us to get at more reproducible measurement with relatively less data. And this is showing like what those actual difference maps end up looking like in the 10 versus the 50-minute data. What we're tending to see is that it's often in areas of transition that we are sort of seeing the most difference, and these differences are quite reproducible across the different amounts of data. Now looking at the individual level, so now we take that, we have that group level parcellation, but now we're looking at our individual level parcellation as well. And we are seeing that we get better sort of parcellation at the individual level with more bootstraps. We see the same improvements in ICC as we would expect based on what the group level bootstrapping results are showing. We see improvements in discriminability. So discriminability is like a multivariate version of ICC. And we see improvements in discriminability as well, though those improvements of discriminability tend to be limited more to the higher number of minutes of data, 20, 25, 50 minutes. So discriminability means like how much can you pick out a person from your data. We see greater within study between sample reproducibility. So this is that those 300 subjects that are cut up into 10 groups. We see more similarity in the parcellation between the different groups there. This is in the subcortical. We see also increases in between sample, between session reproducibility. So now this is looking at the cortical parcellation data set and comparing it to the cortical parcellation from the GSP data set and showing that if you use bootstrapping on both, you actually get much closer alignment overall than if you don't. This is only with 10 minutes of data. We see the same thing for the subcortical parcellation as well. The differences, interestingly, that we see here also are kind of similar to what we saw in the difference maps for the repeatability or the between session reproducibility. And the interesting thing is that we actually see a little dip in the reproducibility of the 50-minute data, and a lot of that has to do with the fact that with bagging, you actually start getting a better fit, a better parcellation. So the 50-minute parcellation ends up looking better than the 10-minute reference or the 10-minute that you were comparing it to. So we see that it seems like actually, though the reproducibility was it was still improved, it just wasn't showing quite as dramatic effects. It looks like it has to do mostly with the pattern of that the 50-minute was looking actually more bilaterally symmetric than the 10-minute was. So the 10-minute, for example, is showing this caudate tail is not different between the two parcellations, whereas in the 50-minute, it was showing that the caudate tail on one side was different. So that's just an explanation of that. In the paper, we discussed this more. This is one thing that I think is particularly cool, I wasn't necessarily expecting, but we also found that bagging has really large improvements in the group to individual alignment. So if we look at the individual parcellation and the group level parcellation, we see that bagging shows really great effects in terms of improving how similar the individual parcellation and the group level parcellation is, which is exactly what you want, because if you were to conduct an individual level parcellation on your subject and you find it were to be really different from your group level, then you wouldn't want to use that group level atlas on that individual person. And then we also demonstrate that this replicates across other parameters, clustering parameters, such as spatial constraint, either with a small amount of spatial constraint or a fully spatially constrained clustering algorithm. And also with different parcellation techniques. So this is using spectral clustering, and we use hierarchical agglomerative clustering in the Ward's criterion base in the actual study. I think I'm running out of time, but I just briefly want to talk about why bagging works, or why we think it does at least. So, supervised and unsupervised learning methods, they suffer from variability due to sampling. And the variability can also arise due to recursive dimensionality. So the idea is that aggregating across multiple resample data sets can provide you a more generalizable model. And because when you assess each of those individual models, each of the models are capturing different elements of the structure of the data. And so even if you were to, develop some amazing algorithm that were to sort of select the best clustering or the best parcellation possible, you're throwing away some other information that are contained in these discarded non-optimal models, right? So like, each algorithm is parcellating, whether it's, based on the data or the algorithm itself is changing. They're parcellating the data from slightly different perspectives, you can think about it. So you get a more holistic perspective. There's, another element that, perhaps beyond nuisance signals such as head motion, et cetera, there's dynamic connectivity that's driving a lot of the variance in connectivity values that might affect how we get better effects, because as we're bagging, we're sort of getting a better representation of that most dominant or central tendency of the data. So in conclusion, we see that bagging improves reproducibility, functional parcellation across scans, sessions, samples, studies, scanners, clustering techniques, and spatial constraint approaches. I think I'm basically out of time, so I'm not gonna really jump into too much of the of my Nilearn stuff, but I just wanted to say that I have really enjoyed teaching Nilearn and Python to people in Brooklyn. At this place, they have these classes and they have this lab. It's like a wet lab, but I'm like one of the people who does more of like the hacking tech, side. And I teach these classes, Neurohacking 101 and 201. And they're most of the people who attend are like, a really big range of ages, different professions, et cetera. But everyone in the public is just so fascinated about the brain. So showing them using, the great documentation and tutorials that Nilearn and Scikit-learn have available, we can actually, sort of show them how to download data and analyze it on their own laptops, which people get very excited about. And obviously I teach them all about all the really exciting projects that are happening in the open science movement, and they're all very excited to learn about them as well. So with that, I'll end my screen share. Thank you guys very much. How should I, there we go. Can I close the screen share?
It's all good now, so thank you so much, necessary groundwork with PyBest the community a big favor. We have questions. If you ask questions for those already mentioned, but if we got one for
Pierre, you're breaking up for me. Should I answer some questions, or are we?
Oh, I'm sorry. Yeah. Please, if
Okay. Okay, I think I heard you say to answer some questions. So I see, given bagging improves reliability more strongly than scan duration, what minimum duration would you recommend? Wonder where to be using BAST, and when would you expect to see diminishing returns when increasing scan duration? Based on what we see in the paper, I think 15 minutes to 20 minutes would be like the sort of the place where bagging hits improvements in reproducibility across the most conditions. But we also show that bagging just at an individual level, so not using any bagging in the time series, but just bagging the across individuals also shows improvement in reproducibility of the population, which suggests that maybe with even less data, you can get improvements regardless of how much actual scan time you have. I see, I believe you said that bagging with shorter scans produces more reproducible parcellations than using longer scans. Could this be because bagging duplicates the shorter scans, making it an apparently more resting, steady state? Easier to have steady mental state for a short period than a long period of time. Yeah, that's an interesting, that's an interesting point. The idea that like variability, I guess it kind of that kind of brings up the question of like, so on what, and I'm not an expert on dynamics, functional dynamics, but like on what time scale are we expecting to see fluctuations in the major patterns here? If it were to be that exact thing that you're suggesting, then we would also expect to see 3 minutes and 5 minutes of data always doing better as well, which we don't. So it does seem like you need at least a minimum amount of data, like signal, so to speak, in order for bagging to have a positive effect. And that kind of also supports what we know from the bagging literature more broadly, that like if you have a little data, bagging is actually going to hurt you because your variability is going to be too great. So that it could be something like that that's happening in those low amounts of data as well. I see, which statistical metadata would you use to evaluate how many resampled data sets are appropriate for bagging aggregation? Well, you see in our analysis that we see even 10, right? So that's one of the cool things. Even if you resample the data 10 times only and aggregate those 10, do you see improvements in the reproducibility? So that could be also something to consider, that we might not need that many actually to see an effect. And hopefully one day maybe we can put some kind of bagging thing into Nilearn or something like that. That would be really cool. In your opinion, could within subject bagging be used to derive more reliable functional connectivity matrices, or is it specific to parcellations? Yes, I think that is a reasonable takeaway from this. I mean, if you are getting a more and that is something that I have looked at a little bit, but I haven't really delved into it. But I do believe that would be the case, that bagging should also lead to more reproducible functional connectivity matrices, because the thing that the clustering is actually using is just a functional connectivity matrix of the supervoxels. So if that underlying thing is more reproducible, then the clustering would be more reproducible as well. But it could also be that it's the variability in how the clustering works itself that is the problem. And so it's the part about aggregating across multiple clusterings that we're getting the improvement in reproducibility. So it's an important question. I do hope that people look at it, and we are looking at that exact point as well. Okay.